Good afternoon, everyone, and a very warm welcome. And it's lovely to have you today here. It is much awaited webinar, which has seen a lot of registration and it has achieved a lot of traction, which is wonderful to know. And without wasting much time, what I'll do is immediately hand over the session to the moderator, Dr. Monalisa Chatterji, who, if I would like to briefly introduce her too, Dr. Monalisa has over 2 decades of experience in small molecules and biologic discoveries and development, and she has pursued her postdoctoral research at Yale University. We all know her, and she's renowned for her leadership across organizations like AstraZeneca and Bill and Melinda Gates Foundation. In fact, she has also been honored by the PSA Office, Government of India in 2022 for her excellence in STEM field. Currently, she serves as the co-founder and director of Sekkei Bio Private Limited and has dedicated her organization to pioneering novel therapeutics for global unmet needs. Dr. Monalisa, the floor is yours.
Thank you so much, Dr. Kavita. I hope I'm audible, and thank you for the kind introduction. It's a pleasure to be part of this important webinar, especially as at Sekkei Bio, we are deeply entrenched into in silico ligand design. As described by Kavita, it is really timely for us as a community of researchers involved and interested in drug discovery to understand the impact artificial intelligence could have and is having on the way we discover new medicines. There is immense hope. There is hope that it will completely revolutionize the way we do discovery, allow us to explore target space and chemical space that has not been envisaged before. It will allow us to meaningfully integrate and interpret big data, ultimately leading to a much higher accuracy of predictive design, and which then brings down the cost and time that it takes to discover new candidate molecules. However, while there is hope, I would put a note of caution. Are we expecting too much from the technology too soon, especially in areas where there is a paucity of high quality data? So in next 90 minutes, what you will hear is you will listen to talks from experts. They will share their thoughts, advancements in the field, use cases, exemplars, and challenges. We hope to learn and then reflect towards the end of the webinar what could be the near-term real impact of artificial intelligence on discovery. So my job as a facilitator, without much delay, I would like to introduce our keynote speaker, Dr. Radha Rangarajan. Dr. Radha is director of Central Drug Research Institute. She has over 2 decades of experience in translational research and product development, specifically in the area of antimicrobial resistance. Her distinguished career includes leadership roles at Dr. Reddy's, Vitus Pharma, and HealthCubed. She has earned many prestigious accolades, such as Biotech Startup of the Year and FICCI Award of Excellence for Women in R&D. Dr. Radha, welcome, and the floor is all yours.
Thank you very much, Monalisa, for the warm introduction, and thank you to the DNDi team for this opportunity to speak here. I am not an expert on AI, but I will offer you my perspectives of how AI can be used in drug discovery and why. So the speakers who will come after me are really the ones who will talk to you about applications and the technological advancements in AI for drug discovery. What I'm going to do is set the context and give you a sense of where I think there is hope, where the challenges lie, and what somebody coming from a drug discovery perspective would like from AI. So I'll start with my presentation. Could I have the next slide, please? The controls are on, yeah, okay. I want to do a little bit of a history lesson here. as far back as human beings have existed, there has been some sort of drug discovery that has happened. People look to nature, people look to plants, people look to cure themselves of their diseases using what they found in nature around them. But when we talk about formal drug discovery, we are really talking about a period that started perhaps in the late 1800s or early 1900s, when people started to think about how specific, very, very potent compounds could be used to cure disease. And Paul Ehrlich is known to have used the term magic bullet to suggest that there could be ways to kill specific microbes in the body without harming the body itself. And in 1910, he identified this compound, also sometimes referred to as compound 606 or salvarsan, which was used for treating syphilis, and at that time hailed really as a magic bullet because it did cure people, even though there were adverse effects. But it was one of those early examples, perhaps one of the first examples of where a small molecule was shown to specifically target a microorganism. Well, since then, the quest for magic bullets has continued, and it's not just in infectious diseases, but also in other conditions where a specific protein or a pathway has become a problem and has gone rogue, and therefore mechanisms are needed to disable those pathways or to downregulate them. Some of the shining examples of the last 25, 30 years are here. Gleevec, this is a treatment for CML, chronic myelogenous leukemia, and it came through a very sustained drug discovery process, and the compound imatinib inhibits BCR-ABL tyrosine kinase, which becomes constitutively active in patients with CML. It was the first example of targeted therapy for cancer. We have statins, which as you know is a very, very widely used drug for controlling LDL cholesterol and for regulating dyslipidemia. It's multi-billion-dollar market, and there are many different statins that are available, and it has brought down cardiovascular complex events for people who are at risk. More recently, there are the GLP-1 agonists for everything from diabetes to obesity. But the history of drug discovery is replete with stories of serendipity and persistence rather than a very, very straight linear pathway to identifying compounds. Next slide, please. I'm sorry, I don't have control over the slides. Okay. So, where do drugs come from? The modern paradigm in which we think about drugs is we understand the biology of the underlying biology causing the disease, and we look to various sources, could be nature, synthetic sources. Now there's cell and gene therapies, and perhaps others as well, which all are harnessed in order to test for efficacy, safety, and identify compounds which have the necessary properties to treat the disease. Next slide, please. Am I able to control the slides myself, Monalisa?
Okay. You can click on the screen, Radha, to get control. Okay. But we are on the next slide. We see your third slide. Yeah.
Yes. Yeah. So the drug discovery process is something like this, that compounds are identified initially as in vitro active compounds, and then they we move to through the process of preclinical development. I'm sorry, can you go back to the previous slide, in which I'm not able to move it with my screen, from my screen. So next slide, please. And the process by which, previous slide, the process by which we get through from a hit to an optimized lead then takes the compound into, please go back to previous slide.
Damon, can we go back to the previous slide, please? Slide number 3. She was still on slide 3. The previous one, yeah. And the next one.
Okay, okay. I'm going to try moving it myself. Apparently, I do have control. Oops. Or there is a lag.
Okay. Yeah, because we do see them moving, Radha.
Yeah. So, oops. I'm not very sure what's happening because the slides are moving on their own.
Uh,
I think there's a big
Radha,
lag between.
mhm.
Okay.
Radha, the control is totally with you. We're taking away the controls. Everything is in your hands now.
Okay,
Try
I'm
it
going to
out,
be here.
please.
Okay, so, we go from hit to optimized lead where through a very iterative process that involves chemists and biologists, compounds, and I'm talking about small molecules, compounds are optimized for activity. And when they are well optimized and safe in animal toxicity studies, then they are taken through 3 phases of clinical development, finally leading up to a new drug application or an NDA with a regulatory agency. And then with the approval of the regulatory agency, the compound goes into the market. And this whole process can take anywhere from, 10 to 15 years. And it can cost a lot of money. And this is what Monalisa was also talking about. Okay, I'm not able to control the slides. So somebody needs to move it to the next one, please. Okay. So what we do know is that for every 100 drugs entering phase 1 clinical trials, about 60 advance all the way, about 60, please go back. I'm having a very bad day with this, with the controls on the slides. Please go back.
Yeah, I'm seeing that. Tamin, is it possible I'll let you know.
Can
you can go back.
Can I share my slides directly? Is that possible?
Of course, let's do that. Let's do that. Sure.
Okay, you can see this?
Yes, we see that.
You can see that? Okay.
Yeah.
All right. So for every 100 drugs that enter phase 1 clinical trials, about 60 advance to phase 2, 20 enter phase 3, and only about 12 gain FDA approval. So this is a very, very staggering number. This is a staggering set of numbers to look at. And the problem really is that not only is there a very high attrition rate, it takes a long time. And the cost of drug development can be as high as 800 million to 2 billion. And even DNDi's own estimates of maybe 60 million or so for a repurposed drug is a very high, which it is a huge cost. And about 31% of that cost is all preclinical development. So I just wanted to give you a very quick example of AMR as a therapeutic area. So this is antimicrobial resistance, meaning that we're talking about bacterial infections. There are about 1.2 million deaths due to resistant bacterial infections every year, and 4.95 million deaths that are associated with bacterial infections. This is 2019 data from Lancet. And when you look at the number of compounds that are in development with antibacterial for antibacterial indications, the number is very small, 64. I just told you what the attrition rate is to go from phase 1 to phase 2 to phase 3. So this number is not going to really get us drugs in the foreseeable future. But it's not for want of trying. If you look at these references, which I put for you here, the one at the very bottom, drugs for bad drugs, this is a summary of GSK's experience where they did 70 high throughput screens and did not get any candidates. Escaping the Labyrinth, this is AstraZeneca's review on 65 high throughput screens that did not get them to a clinical candidate. A completely different approach of trying to find antibiotics from soil bacteria. This resulted in a compound called tixobactin. Again, never made it, or its development has not been reported. And more recently, a high throughput screening by GARD-P of small molecule libraries found 2 hits out of 48,000 curated compounds, but none that are optimizable. So, there are some very serious challenges from a science perspective, identifying good compounds. And when compounds go into clinical trials, they still fail. And that's because, there could be issues with efficacy. There could be issues with safety. Efficacy is the number one reason, followed by safety, and other reasons such as commercial. opportunity. So the magic bullets have been elusive, and people continue to need much better techniques where predictability, cost, and time are addressed. We're able to address clinical unmet needs of subpopulations, and we're able to enable drug discovery and development in low and middle income countries. So can AI/ML be an answer to this question? So traditional models of drug discovery are sequential and lengthy, and ML uses algorithms to parse data. The algorithms are able to learn, the algorithms are able to use vast amounts of data to enable greater precision. And they are useful, these algorithms are extremely useful when a number of different variables need to be considered, and the approach is more integrated in the sense that you're not optimizing for one feature at a time, and it's less sequential. So there is a lot of promise in this method. And there have been some early signs of success. This is an example of a compound that the Broad Institute published about. It's called Halicin, and it came out of a sequential a mix of testing compounds from a library, generating the data to build the algorithm, and then testing large in silico libraries to identify this compound Halicin with broad spectrum activity. Then following that, they were able to show that the compound was active in a topical model and in a Clostridium difficile diarrhea model. So both in vitro and in vivo efficacious. Similarly, there's been another example to identify compounds through a process again from the Broad Institute. This is targeting Acinetobacter, one of the major pathogens of hospital-acquired infections. And these are all hits. They are leads, but they are not drugs. And there are many reasons why they're not drugs, but they give us hope that it is possible. Here is an example of a company called Benevolent
Rather
AI.
a 12-minute checkpoint, time check.
Yeah, since I had a lot of audiovisual
Sure,
issues.
you get, sure.
Yeah, give me a couple of more minutes. In silico medicine, which is developing a compound now in phase 2, and all the way from zero to something in phase 2 or first in human phase 1 studies in 18 months for idiopathic pulmonary fibrosis, a very large unmet clinical need. So there is promise, and various institutes and both academic and industry are harnessing AI/ML for drug discovery. CDRI is no exception. We have, in fact, a collaboration with Dr. Gaurav Ahuja to identify inhibitors of NDM. We also have a collaboration with a company called Shravati AI to identify PARP1 inhibitors for cancer. So the major problem in drug discovery is really identifying chemical starting points. If you look at this graph here, it's clear that, especially look on the right, proteins for which chemical probes are available, a lot more studies are possible with them than those for which they are not. And perhaps because of that, a number of targets that have been put to use for drug discovery have been proteins for which there has been a starting point, like kinases. So in terms of the future, what I think are the priorities, we need to look at AI/ML as a resource, as a tool to make the drug discovery process more efficient. Data can be heterogeneous, often not publicly available. So we have to generate data. Oftentimes that comes as a cost. Can we have data sharing models? We need to go beyond binary models, meaning that we can't simply be building algorithms to identify compounds which are efficacious. They must integrate all of the aspects of a drug, such as ADME, safety, and so on. And ultimately, I think we need to think about whether we can create OpenAI for drug discovery. Otherwise, we're going to be reinventing the wheel over and over again. There is huge need for computational resources and interdisciplinary teams. And the challenge to AI/ML researchers and developers is, how to make these tools more accessible to all. So I'll leave these questions for our esteemed speakers in the panel to dwell on. And thank you very much for your time today.
Thank you so much, Radha, and apologies for the initial hiccup. Technically, we often do face challenges, but What you did is you showed us the challenge and the daunting task it is to discover a new medicine, the cost and the time that it takes. But you have also given us hope and shown us few glimpses of successes that are coming up. With that, I would invite our first expert, Professor Sastry, former director of CSIR Northeast Institute of Science and Technology, Jorhat, Assam. He brings extensive expertise in interdisciplinary research spanning chemistry, biology, modeling, and informatics, pioneering efforts in leveraging artificial intelligence and machine learning for Industry 4.0 and 5.0 concepts. He is notably recognized with prestigious awards, including the Shanti Swarup Bhatnagar Prize in Chemical Sciences and the National Bioscience Award. Before I hand over the floor to Professor Sastry, I hope that we would have Radha back at the end of the webinar, and we could get some of her thoughts. In addition, I encourage the audience and the viewers, if you have questions, please do populate in the chat box. If we can and we have the time, we would definitely take your questions. Thank you so much, and over to Professor Sastry.
Good afternoon. Thank you very much, Monalisa. And my job has become a little easier by the introductory speech by Radha Rangarajan. I just wanted to make it very simple. And definitely, I'm not going to put in a lot of data, but I wanted to put some ideas that how this, can I have my slides? The revolutionizing drug discovery, pharma, and healthcare. So basically, we have drug discovery as a necessity to confront the problems that we have in terms of the diseases. While pharma is a pharmaceutical industry, to make things straight, they would be looking forward to make money by using this as an industry, and healthcare is something which is in between. And I just wanted to tell you that we need to think about how a new technique like artificial intelligence will play an important role. Before I go to my next slide, I would like to tell that the laboratory's name that I have kept is the Fifth Paradigm Lab. And I always feel that when a new paradigm comes, we should know how effectively to use it. As most of you know, that experiment and theory as the first and second paradigms of science, and they played a very important role in drug discovery. When modeling has come, this third paradigm has played a particularly important role in drug discovery in the form of molecular modeling, molecular dynamics, and lots of simulations and mathematical modeling. The fourth paradigm is data science, artificial intelligence and machine learning, where we have a huge data, and that data will be useful to convert into knowledge, and this knowledge will be useful in solving the drug discovery problems. Fifth paradigm is something which will be effectively integrating all four paradigms of science. I just wanted to tell you that in my talk, I wanted to make very simple introduction to the concepts. Like if you look at Oxford Dictionary and then see what is intelligence. Intelligence is written as the ability to learn, understand, and think in a logical way about things. The ability to do this well. This is intelligence. So the ability to learn, are humans good in learning and understanding and thinking in logical ways? This is something which is very, very interesting to see. If somebody does it well, they are called as intelligent human beings. So when you look at artificial intelligence, it's written as the study and development of computer systems that can copy intelligent human behavior. But I think it's a skill set. When you want to learn things, definitely a computer can learn much better than any human being. Understanding, I think so. A machine can do it much better. And logical way about doing things, and probably a machine can do it well. And this has been proven long back when the computer has won the chess game against one of the grandmasters. Most of the grandmasters, now they cannot play with the computer. So while we are able to learn, understand, and think in logical ways, the ability to take decisions or the wisdom is probably sometimes beyond the reach of the computer systems. And then we will have to see that how the computer will be different, how effective it is compared to human being. And at the same time, we also have to know how human beings are very different from computers, how good they are. For example, if you want to look at a technological adoption, I have given some examples in the left side, like electricity, which took a lot of years for people to adopt. Even about 60, 70 years before, most of the Indian villages did not have the electricity. Even in the world, it took a sweet 50 years before we can adopt this great technology. And when we look at the telephone, radio, television, it took a lot of years when the discovery was made to come to the mainstream. However, when you look at the personal computers and mobile phones, the web and the smartphones, the way in which these new technologies are coming at a pace where the adoption, we don't have time. For example, ChatGPT has come. It's about a year old. These pre-trained transformers were so fast and they don't give you enough time even to adopt. In the past, the technological promotions like changes were intergenerational, and society, people, these productive systems could adopt slowly, and there are positive workforces and that could balance in the long term. Whereas the current plethora of new technologies, the new apps, new webs, new things that are coming are intragenerational, and society, people, and productive systems have no time to adopt. And in contrast to the positive workforces, I would probably try to say that these are negative workforces in the short term and no proof to be balanced in the long term. So the way in which the systems are adopting is too fast. And now let's come back to drug discovery. As has been mentioned in the previous speakers, I just wanted to say that there are different areas like drug discovery and development. Basically, you need to have a molecule which will show some efficacy against a particular disease. And that's probably we can call it as a lead. And then we have to optimize that lead to have enough confidence for that molecule to be put for making money. So we have to get an IND application, investigational new drug application. Then the clinical trials will start. As Dr. Radha has mentioned, the clinical phase 1, phase 2, phase 3, and the attrition rate is so high and the costs are also very high. So when we go from the drug discovery to development, there are large number of challenges that one has, and experimental approaches, random approaches, very, very controlled approaches have played a very important role. And we have other aspects, especially the precision medicine, which has become very important. I will try to discuss about it in the next slide. But one of the most important aspects that has come up recently is drug repurposing, which played a very important role. That there are many, many drugs which were discovered and developed and got an FDA approval, and afterwards probably they were not being used, or they were used against a particular disease. But these drugs appear to be very effective against a completely different disease. That identifying the new uses of existing drugs by analyzing large data sets and biological knowledge, where artificial intelligence is playing a very important role. And supply chain optimization is extremely important. While we can try to have the drug discovery development processes are there, but to scale it up to a large scale, the supply chain optimization is very important, and we have witnessed how important it is during our COVID times. Various countries which depend on the APIs on other nations had a huge issues with this. And the most important aspect is regulatory compliance and safety. The safety versus risk ratio. And while we talk about a large number of uses that are there, we also have seen some of the catastrophic results like thalidomide or statins and many other over treatment of a particular disease lead to a lot more complications than actually resolving problems. So AI algorithms can analyze the real world data and try to very effectively optimize the risk versus benefit ratio. And clinical trial optimizations, drug formulations and delivery, the drug release kinetics and absorption profiles and formulations, and how the enhanced efficacy and targeted delivery, they play a very, very important role. And artificial intelligence, machine learning, deep learning, symbolic AI, IoT, many techniques have played a very, very different role in this game of drug discovery, and it has provided a good arm to deal with this complicated drug discovery in a very interesting fashion. So I see Dr. Monalisa raising her hand. I have a few. So I will try to complete this. Delivering 4P, the personalized, predictive, preventive, and participatory medicine has become absolutely unavoidable. While most of the people would like to do that, the data is huge. Humongous amount of data is there. And I would like to actually find out that this particular data sometimes is very divergent. And many of the indicators are working in concordance and sometimes in discordance. And it's very difficult to come up with a particular result or a particular decision which will be applicable to all the population in a general way. So it's extremely important to see how this personalized medicine will play an important role. And artificial intelligence in drug discovery has actually radically transformed data architecture and data sharing, and has improved the accuracy, predictability, and speed of drug discovery. And drug discovery based on new skills and talents and new geographic distribution of AI for drug discovery is very important. And it's all about finding the right molecules and finding the right targets. And machine learning, as it has been shown in a large number of cases, actually you can see a number of papers. Even we have utilized machine learning in addressing a large number of issues in finding out the right molecules or finding out the right targets or right protein-protein interactions and how mutations can play a very important role. And this has become a very important tool in the drug discovery in recent years. I just wanted to tell you, so just I will probably discuss about Industry 5.0 very quickly, because
Dr. Sastry, 1 minute. You're on the 12-minute time.
Good. So we just have to go from the Industry 4.0, which is about IoT, cloud computing, big data, and cyber physical systems. They themselves are playing an important role, to Industry 5.0, where human-centric solutions, customized, personalized, and real-time access, they become very important in drug discovery. I just wanted to leave it open that there are a large number of new techniques which have come, and they might probably complicate things or simplify things, but they become very important in sort of impacting drug discovery, pharma, and healthcare. It may be revolutionizing, or it may be changing or metamorphosizing, and we cannot ignore it now, and then we will have to somehow see that how to effectively use them. Thank you very much.
Thank you so much, Professor Sastry. You've given us a breadth of applications or impact AI can have, as well as the technologies that are being developed in parallel. There is one question, sir, for you. Quickly, if you can. So there's a question that's saying that are there data sets or is data available, or do we need to build data sets? So this is a question on to use AI methodologies. What is the source of data?
So basically, there is a lot of data that is available in chemoinformatics. For example, if you look at the data that is there, even you take Campbell database, or you can take, there were 30, 40 different types of databases. And we developed the database called Molecular Property Diagnostic Suite. The chemical data sets are huge, and the information is very huge. And there's a lot of scope to derive a lot of information. This is like a very, very interesting area. A lot of data is available, and we have too much data and too less knowledge because we are not able to process it in effective ways. And AI machine learning is definitely to be employed in the area of chemoinformatics and small molecules discovery, as well as in biomolecules and peptides and peptidomimics. There's a lot of data, and the velocity with which it is accumulating is very, very high.
Thank you, Professor Sastry. One more quick question and a brief response. There's a question that can we use artificial intelligence for prevention of diseases or diagnostics?
Absolutely. This is very important because now the immunomodulation, inflammation, and I feel that the healthcare and medical care are completely different. When you try to see the disease is a perturbation to health, and there are many ways in which people are trying to restore the health. If I can quickly say that health is a complete physical, mental, social well-being, and not merely the absence of disease or infirmity. And physical, mental, and social well-being, this has to be balanced. And now people are also adding emotional and spiritual well-being. So now when there is a disease, whether you treat the disease or restore the health, immunomodulation, immunocheckpoint therapy, anti-inflammation, nutrition, they are all playing a very important role. So the holistic way of understanding is very important, and prevention definitely, improving the health, immunity boosting are becoming more acceptable, more relevant, and more trendy, especially after the COVID.
Thank you, Professor Sastry, for your presentation, sharing your thoughts. I also want to thank all the people who are putting in questions. Please do keep putting them in. We will move on to the next speaker, but we'll try to take them as we go through the webinar. So our next expert is Professor Shandar Ahmad, a professor and coordinator of DBT-Bioinformatics Center at Jawaharlal Nehru University. He's renowned for his pioneering work in bioinformatics and computational sciences. With extensive international collaborations and groundbreaking research at CSIR Lab, Professor Ahmad has become a leading authority in developing cutting-edge algorithms for biological systems and advancing deep AI and ML technologies. His presentation is titled as Use Case Scenarios of AI in Early Stage Drug Discovery. Professor Ahmad.
Thank you very much. Can I have my slides, please? Can I have the control? Much for the organizers for having me here, and it's wonderful to listen to two of the experts in this field in the drug discovery, and it sets a very right background to where we can talk about AI specifically. And in my talk, I'm taking it to a little closer to AI and probably less close to the drug discovery process. In the process, the talk I'm talking about AI in general. So introducing my lab first. My lab is called Sci-Y Lab. Sci-Y, as it says, the question of scientific questions for driving happiness, but it also Sci-Y in Japanese means happiness itself. So we try to draw happiness from our scientific queries in our lab. And my team consists of 3 groups. One is, I think, but the sides were not visible. So 3 groups. One of them works on the systems level drug discovery. One works on the genomics aspects of biology, and one works on the computational algorithm. Come from computer science background. Putting this in perspective, my talk has been targeted at more like people who are already working in the field of drug discovery. So from that perspective, you can think of AI in drug discovery to be used either as a commodity. So as a researcher who is active in drug discovery, for them, how should they look at AI when they want to utilize this in the process of their drug discovery? For them, AI can be treated just like a commodity. Like in this slide, I show you that a child can play with a device which has been made on AI. So for the application of AI and the tools that have been built on AI, while you're trying to use these tools, you really don't need to know AI. For you, AI is just like any other technology. If you are doing, say, structure prediction using an AI-based tool, you don't need to know AI yourself. Somebody who knew AI has already incorporated that into that tool. So some of the tasks that are done with this is structure and function prediction, affinity and docking score predictions, toxicity, adverse effects. So these are the questions that have already been raised in the previous talks about what are the questions of drug discovery, where the bottlenecks lie. So for this aspect of utilizing AI, we do not need to probably understand much of AI. What we need to know is what are the tools and devices which are available and how well they perform, and what are the limitations, just like any other piece of technology. On the other hand, for novel problems and asking questions that have not been asked before, or looking at the scientific question of drug discovery in a way that has not been looked at before, or to try to advance our tools and techniques, we need to think of AI as a method which will substitute your current research method by a data-driven question. So we, as the chemists, biologists, and mathematicians and physicists, we have been used to solving problems from the perspective of fundamental mechanistic basis of something. Whereas AI tries to do away with the mechanistic basis and tries to find answers without really looking into how this answer was found. So you have to transform your thinking from thinking of just finding a solution from first principles, finding a solutions from the way we understand how it works, to a data-driven approach where how it works is less important than how well it works. So the novel questions for this type of AI thinking, for this type of an application of AI in your drug discovery, you need to understand AI. You need to understand what are the possibilities. You have to understand the language and the skill set to train data-driven models. So these are the two distinctions. These are distinction I would like to make in 2 different scenarios of AI usage. And I covered some of them, the cases of in which the AI has been used, like AI as a commodity. Some of the use case scenarios can be biomolecular structure prediction. So structure-based drug design is is a very established standard technique that we want to know the structure of a target. We want to understand how it folds, where the binding pockets are, what are the structural properties of this. So when you have earlier you were doing your homology modeling, you were doing with say the ab initio or the first principles. Now you have AlphaFold. So probably when you want to use AlphaFold or ESM fold or Rosetta fold, the list of tools that have been written here, and some of the tools I have added is Dynastic and ASAView, DBS, PSSM that we have developed in our lab, which talk about DNA structure from sequence and trying to understand the dynamics of an entire genome. So when we try to use them, we don't really need to know AI ourselves. Docking and affinity prediction scores, when we try to, then these tools are available, deep docking, DiffDock. These tools, again, all the list of these tools that are listed here have been developed based on using AI. But while using these tools, you don't need to know AI, because somebody who developed these tools already knew it and has already packaged it with the software they're providing. For the protein design, you have a sequence you want to know a particular protein sequence that will fold in a particular way or perform a particular function. These are the tools, ProGen, EvoDiff, and probably many more, which I have not covered here. So biomarker and target prediction. So we starting from just the discovery of a disease, and then we try to know biomarkers and try to know what targets would probably be usable. So this is probably an intermediate level where some use of the AI will require a little more understanding how we use, because these are not ready-made tools where you can directly put some data and get an answer. BioGPT, ChatPanda GPT, KG, where you can ask questions in a natural language and it will answer you. Just the ChatGPT answers you the common language questions. You want to do mutant analysis, particularly for personalized drug design, drug resistance, etc. You have deep docking software, DiffDock, etc., which provide you these scores using AI. And when you want to look at the systems level, we heard about we often about attrition rate of drug discovery is is so high. So we want to incorporate the systems level impact into the drug discovery process at a very early stage. And if we can do that, that would be wonderful. Our attrition rate can be reduced substantially. And that is where this data connectivity map, and this is perhaps I will talk about in a specific application that we are trying to work on in our lab. So the tools like MPGEM, which we have developed in our lab, Jigyasa, where we have integrated the biological data, GEO, to perform meta-analysis and save a lot of time for the users. And when you look at, when we say thinking and modeling of AI, then what you have to do. First part I said, when you use AI as a commodity, these are the names of the tools that you have to know. What is possible you have to know. But when you want to model yourself, when you ask new questions, you have to know the data-driven modeling, the questions, and you also have to know that the AI and ML does not really bother about the means, and justifies the mean. How you reach that answer does not really matter. But what still matters is you should be able to explain your models later. That is why the whole area of interpretable or explainable AI has come. But then in order to apply them effectively, you have to understand whether the models have been generalized enough, whether the relationships that you are discovering have a causal relationship or just a correlation, whether the model that you have selected is effective in a particular problem that you are dealing with, the data augmentation problem, somebody has already solved these problems for a similar data. Can you utilize them if you do not have sufficient data, and how to use the pre-trained model? So these are the questions that you have to understand, where you use AI relatively at a lower level and closer to AI, and instead of just using it as a commodity. So I take here now some examples from our own lab. We looked at this data from Gene Expression Omnibus, and we see that a large number of experimental data, transcriptomic data is available. 220,000 series, independent experiments, which have been performed on 25,000 gene expression platforms, and which is about 7 million experiments at the time when I built this slide. Now, many of these experiments provide you some expression data for 15,000 genes, some provide for 20,000 genes, depending on what platform was used. Many of these experiments are incomplete. And that is where we have tried to integrate so that people, suppose you want to know a disease signature and you are looking for what drugs would possibly reverse this signature. So you want to collect all the information that is available in this GEO and perform a meta-analysis. But meta-analysis takes a lot of time, how to integrate this data around different scales, data around different entities, their different disease signatures. So we have actually tried to integrate them, and for 4 of these 3 platforms that I'm showing here, the microarray platforms, we put them together, we harmonized them and brought them to a same scale. And also the data, the genes which were not present in the smaller platforms, we have predicted using an AI ML, we have predicted the expression values for them. So this works well, and this leads to what we call the multi-platform gene expression matrix, so that if you have to utilize this data from GEO, carry out a meta-analysis, you simply extract the names of the samples and just move to the next step instead of data pre-processing. And after that, here is an example of TLR5. For example, if we want to collect, we were looking at one flagellin impact on TLR5. So we wanted to see whether we can design some small molecules which can have the same perturbations as flagellin does to TLR5. So for that, we can collect this metadata from this MPGEM, large data for meta-analysis from MPGEM, and create a disease signature and utilize this. A tool that I would like to explain here is the CMAP that has been there for a few years now, but has not really got that much of traction yet. But this is a huge repository of the gene expression changes that drugs cause. So about 1 million profiles, a large number of perturbations like these drug candidates, they have been tested. The power behind this is that this utilizes the gene expression changes. It collects the data from just a small set and then predicted it using deep learning for what will be the expression value for the rest of the genes. So using this data, combining these two, we are able to, since I'm already running out of time, the outcomes on these experiments on TLR5, we were able to, I think my text is missing from the slides because of the color change. Basically, with the help of AI, we were able to show that if we collect the first leads using CMAP, creating the disease signature from our own tool, MPGM, putting them together, we are able to validate them. Once we collect the leads from CMAP, then see whether the leads that have been generated are also structurally valid. Are they really valid in terms of the fragment-based signatures? They were very confirmed to be very robust leads, and but putting them all together using AI is really a challenge, and this is a area which we are trying to simplify for users. So concluding remark finally is that most computational drug discovery tools have moved. So everything that you used to traditionally do structure prediction, docking, all these experiments, more and more of those tools are AI driven, but you really don't need to know about the AI behind them except for a general interest and try to use it more effectively. But first principle applications are needed to create novel questions, novel avenues, and asking, for example, if you have a huge gene expression data, asking questions, what will happen if I knock out this particular gene? So asking questions of this kind is not possible currently from any data, but AI-based tools and techniques in which we are contributing slightly will probably be able to answer such questions. That is my final thought, and I just express my thoughts. Thanks for that. Sorry, I had to rush.
Thank you so much, Professor Ahmad. And I think seeing a real life example from your lab where AI allows you to integrate data sets, which probably would have been very challenging if you did not have the tools, if I understood correctly, is really to see it in works. There is a related question, which I would extend it a little bit. So the question actually states is, how do you see that AIs, how can we utilize AI in tox predictions as well as in the space of ADME? But I would extend it a little bit. When you visualize utilizing artificial intelligence, especially in data-rich areas, how do you think it impacts efficacy, safety, and as well as PK or ADME properties? A general overview.
A lot of work is being done on this. So the first, what we are doing with the help of CMAP is that any drug candidate, we know the expression level changes that it will bring about in every tissue, in the listed tissues. What will happen to the liver? What will happen to the kidney and different tissues? So this data is available. So when you have a candidate drug in the beginning, you already know something about its toxicity, ADME, those properties in terms of the gene expression signatures. So cellular impact of drugs is incorporated right at an early stage when you are looking for the candidates. This is how it helps. And secondly, the predictions at a later stage can be carried out if we have the one area in which it is very helpful is species translation. Many a times the drug attrition also happens because the drugs work on animals, but they don't work on human. So how to accurately translate the data from one species to other using simply a data-driven approach. That is also an area in which a lot of people are working, how the phosphorylation changes. So without knowing exactly which kinases and what is happening at the individual mechanistic level, you can predict the impact at what we call systems level. And systems, the entire system is much easier to work with when we work with AI, because the understanding them from the first principles requires really a large number of parameters, large number of dimensions. So many hypotheses to be tested and evaluated, which automatically are taken care of by AI-based models.
Thank you so much. Unfortunately, there are more questions coming in. Thank you, Dr. Sastry, Professor Sastry. You're already answering some of them in the chat. If there are questions that are related, Professor Ahmad, there's questions specifically for where the data was collected from your lab. If you could take a look, that will be helpful. I would be moving on to our next expert, Dr. Gaurav Ahuja. He's Associate Professor at the Department of Computational Biology at IIT Delhi. He leads a pioneering lab integrating artificial intelligence, chemistry, and biology to understand the functional significance of endogenous metabolites in health and disease. His expertise in chemical biology and chemoinformatics has earned him prestigious awards, including the Embo Young Investigator Fellowship and the Ramalinga Swamy Re-Entry Fellowship at DBT. Dr. Ahuja's presentation is titled Challenges, Hype, Hope, Reality, and Success Stories in AI in Drug Discovery. So looking forward to it, Dr. Ahuja.
Thank you, Monalisa, for the nice introduction and summarizing some of the work which we have been doing. And also thanks to the other organizers for having me here. So I just need this sharing. I just want to share my slides. Is my slides are visible?
Yes, we see them well. No problem.
Okay, good. So thank you once again, guys, so for having me here. So my name is Gaurav, and I'm an associate professor at IIIT Delhi, and I'm also a core member of the Infosys Center for Artificial Intelligence that is too hosted at our campus at IIIT Delhi. So as Mona Lisa just said, like I will be talking about the challenges, some of the hypes you have been seen on news all the time. You know, this company has got 3 million funding, 10 million funding, and so on. AI is doing this, AI is doing that. So is it really hype, or there is something it eventually leads to? We will be talking about the hopes as well, the reality. We will do a stringent reality check, and I will be ending my story after talking so much negative in the beginning. I will be sharing some of the successful cases what we have up to now using AI in drug discovery. So to start with, I mean, I think many of my former speakers over here has already, talked about the role of AI in the drug discovery cycle. Let me quickly talk about it. So if you can nicely and comprehensively see, AI has been in every place now. The more and more time the companies are trying to use AI everywhere, not only in the drug discovery, but you can see the chat GPT is one of the thing which all encountered. Now even if you can go to any hospitals, like we go to Rajiv Gandhi Cancer Hospital, even in AIMS New Delhi, many of the departments, I don't say it's been adopted at a holistic level, but many small departments, individual doctors, every of them are using one or the other solution where AI is involved. So AI is definitely is one of the core of doing the accelerated drug discovery. But nevertheless, not only in the drug discovery part, but people have also been thinking and executing. Can we also use AI for the clinical trial optimization? Because if you have seen some of the previous talks, then you know that it is only the clinical trials which take the maximum of the time, near about 6 to 8 years. So can we optimize the clinical trials and try to expedite? So if the clinical trials are perfectly optimized, the overall drug discovery cycle will be squeezed, and we will be able to do things. We don't even have to wait for 10 to 12 years for a particular drug to bring it to the market. So this has already been, thought of. Now moving a little bit more forward, people are also using AI in developing diagnostic tests for personalized medicine. For instance, there are a couple of companies also started within India, also foundation. One is one of the companies in the oncology space. They are using AI and the genome sequence, and they are doing predictive biology to find out which of the drugs can work for a particular cancer type. And there are many other companies that are like there, so which are doing, and even we have a very nice Indian company called Data Genomics doing wonders by integrating AI in personalized medicine. And last but not the least, we have a medical imaging analysis. People, as you might have seen very recently from Pichai, like from Google, like they are looking into the retina of your eye and trying to predict the abnormalities happening in your body. So that's, and, people are, building models by taking a lot of MRI data scans, lot of X-rays data scans, so that you are not, the radiologist don't need to spend hours in reading one report. You can first feed it into the AI model, and the AI model can give you some recommendation. So these are such, some of the four major domains which people are integrating AI in the drug discovery cycle. Now, what are the key challenges these pharma companies have been facing? I think many of them has been talked about before. So one of this is time and cost. So this time and cost comes hand in hand, because bringing one drug to the market is more than $1 billion. Recent estimates are around 2.5 billion per drug to the market, and it takes around 10 to 15 years for a drug to bring. So this is one of the most important challenge pharma companies are basically suffering from. Now the other thing is resistant disease. For example, there are certain diseases for which the conventional drug development cycle cannot work. These diseases are very endemic to some particular things, so it is not the profit making. And there are also certain diseases which basically require, like something with the HIV, For example, we know that the antigen keeps on changing, so there are no effective drugs. So you need to modify drugs as the disease progresses. So this is also one of the other problems. The third problem is the trial success. Like as former speakers rightly mentioned, 1 out of the 10 drugs which goes into the clinical trials, they get FDA approval. So the success rate is 10%, and this 10% success rate is counted for the ones which undergoes clinical trial. So there is also a filter before the clinical trials. So majority of the drugs shoot down at before the commencement of the clinical trial. So the success rate is really minimal. And there are other challenges, basically, for example, selective drug molecules or the desired targets are inefficient financial incentives tied to the market size. So moving a little bit more problem and more technical things into challenges in AI. As my former colleagues and my mentors have already mentioned, the data is the key essence for AI. And it is very well known, the garbage in, garbage out, the good data in, the good models out. So models are, you can build a model. So if I talk to you about technical, in the technical jargon, you can build model in like 5 minutes, depending upon the size of the data. If you have very relatively small size, it is just 2 lines of code. You can put any classification algorithm or regression model. But the point is, you need to ensure that the data quality is good. The data is available. If you really ask me, while working for the last 5 years in India on drug discovery or related fields, I can tell you that there is a lot of data. I agree. The data is flooding all over the internet. It's true. But there is a dearth of good data and good annotated data. Let us talk about Cambial. Am I right? So Cambial is one of the most carefully crafted and assembling the information about drugs. But the point is, if you go and deep into the Cambial information, you will see that it has experimental information, some of the information coming from indirect measures. So if you need to have a particular domain knowledge in order to extract and filter even the Cambial data set to get the real gold out of it. So, the data quality and quantity is one of the biggest challenge. And many pharma companies, they do have money, expertise, they do a lot of, essays, but they never put their majority of them, they don't put their results outside. Because just to remind you, a good model requires positive data, which means the drugs which have some phenotype, and they also require negative data. So these pharma companies, if something doesn't work for you, you throw away the data. But as we already know, every data, even the negative result is a result. So the negative data is also important. But there is a dearth of good quality data. Both their quality and quantity is challenging. And as you can see the recent reports over here, the reproducibility, people have taken multiple challenges. There are a couple of papers people have gathered from the public publications, and they try to reproduce it. And in most of the cases, the reproducibility is a big issue. And even for the connectivity maps, there is one documentary out. So people do mention that the reproducibility, especially when you generate a gigantic amount of information from a multiple high throughput platforms, the reproducibility is always a concern. And if your data is non-reproducible, it means that you are feeding the wrong information to the model, and therefore the model learns wrong, and therefore the predictions are wrong. So that is the first level of problem. The second level of problem is the complexity of the biological system. As we already know, starting from the very basic, the central dogma of molecular biology, DNA to RNA, RNA to proteins, and so on, we know that the biological system is highly complex. And now people have recently found out the way to represent the biological system as a holistic view by generating these knowledge graphs. But the another level of the problem is these knowledge graphs are effective, and there are a couple of successful cases where you take the 100 years of literature, biological literature, convert them into nodes and edges, and connect and make a very big graph representing the entire knowledge of the biological system of humans. But one of the critical problems with them is the computational requirement and the authenticity of the data, because all these knowledge graphs are built on the publicly available information. And as I already quoted before, in many of the cases, the reproducibility of the results are the major concern. Now, the third thing is integration into the existing framework. So there are two thoughts, the pharma companies, and there are two school of thoughts. The first school of thought is the existing pharma pipeline can integrate AI at each of the existing thing. And the other thing is you remake and make an entirely new de novo workflow, which is basically entirely integrative AI in all of the subsections of the drug development. Now, the thing is, the pharma companies, the one of the elegant way to do that is to integrate AI at different steps of their standard workflow. And this integration is also posing a certain challenge. And again, there is also a regulatory and ethical concern. If your predictions are wrong, if money being spent, and if there is nothing came out of that, then who is, if the predictions are wrong, for example, you have AI model predicted you to be suffering from cancer despite you are having no tumor in your body, then who is responsible? Is the responsibility goes to the person who has made the AI model? Is the responsibility goes to the algorithm itself, or is the responsibility goes to the company? So there are many regulatory and ethical concerns associated with this. Now, that's sad, but at the same time, if you can see this very nice internal report from Arcturus, then you can see that if the y-axis is the cumulative companies and on the x-axis is the time frame, and now you can appreciate that in the past few years, the companies which are doing hardcore drug discovery and implementing AI and ML, you can say there is a real surge increase in the number of such companies, which really means that at least the pharma industry or the drug discovery industries, they recognize the importance of AI in drug discovery. And many of these companies, new startups, as well as the old pharma companies are adopting AI at an alarming rate. One thing, majority of the new startups are basically academic professors or academia converted startups, but still there are some successful stories I will be talking in the end of the slide. Now, all of the recent successful or partially successful companies are working at two different axes. So you can see on the y-axis there is a focus on life science data mining. So there are hardcore industries which are putting more stress on data mining processes, like gathering the scientific data, put them into a structure thing so that they can train their model, something like what ChatGPT has done for the generic data. Now on the x-axis, you can see there is something, the life sciences, they are doing automations, like you need robotics. You need robotics to, let us say, generating a new strain. So you can see that the companies are divided into four quadrants. These are the companies, I could say, really playing at the cutting edge. They are mining the data from the internet, and at the same time they are also doing automations for experimentation. And therefore it is a very good news for us, like drug discovery evolution being happening, and AI is getting into the DNA of the futuristic drug discovery. Now, what is the
Right.
hype? As I said,
One more minute, your 12 minutes are up.
oh, sorry, let me accelerate. So, hype around AI, you can see that after years of hype, the first AI designed drugs fall short in the clinic. This is something disheartening. And there are a couple of other reports you can see that. One of the classical problem is even we fail. Many pharma companies fail to share the failed results, which led to the critical problem. And the hype is already going on, but there is a requirement for the reality check. How much is the AI is contributing to this acceleration? So I just want to quote something from Patrick Straub, which already said that the benefit of using machine learning can be evaluated, and you can do that by doing at least a thorough power analysis with 150 clinical trials to observe a statistical significance. Is in reality implementing AI in the drug discovery really helps, or it doesn't help? That analysis needs to be done, and at least for that we need, if we want to get a power of 80%, we need to conduct this analysis on 341 samples. Benevolent AI, I think my next talk from the speaker will be talking in detail about benevolent AI. I just want to tell you, this is one of the most successful young company from the UK, and they are doing wonders by using AI knowledge graphs as well as all the recent algorithms used in AI. Now, somebody has rightly said, can we integrate ChatGPT with drug discovery? Yes, people have thought about it. And CanCrow is a standalone tool, again, developed from academia. You can try it. You can help in designing the chemistry tools, molecule tools. It helps you to fetch the literature. Many things you can do, something with integration of chemistry and ChatGPT. Now, this is something what my lab
Dr. we
basically
really have run out of time. So if you may summarize, that'll be helpful. Thank you.
Okay, I summarize on this slide. I just want to show you the power of AI. This is the work which recently we published to identify the molecules which can, to identify the anti-aging compounds. Just wanted to show you, we have screened about 1.1 billion compounds being sitting in Delhi, identified some of the geroprotective chemical space, the novel molecules which have never been reported to be anti-aging. We took these molecules, tested on the human published data, as well as did a couple of analyses on yeast, C. elegans, and human fibroblast, and really proved that these AI-based anti-aging drugs really do wonders. So this is something I wanted to show. AI provides you the speed, and the speed is to screen 1.1 billion compounds. Thank you so much, and I'm open to questions. Sorry for taking some time, extra time.
No, thank you so much. And my apologies, because I have to make sure that our final speaker has the right time to go through. But it's exciting to see the hope that you have given us and the examples. If I may ask a very quick question, that is, you mentioned good quality data. So is the only solution is to generate good quality data, or there are ways of addressing variable data set during your modeling time? So can you address the variability that is there in the data when you model, or you have to go back and generate good data?
I think it depends upon each and every companies or labs. Like if you have money, you can always do high throughput screenings and all that. But the ideal way is to have a hardcore domain knowledge, try to take into account the domain knowledge and try to take public data first, try to narrow down, and you also generate your own data and put it as a testing unseen held out data set so that you can know how good my model is performing. So I think a combination of both is the way to go.
Thank you so much. So moving on to our final expert speaker is Dr. Charles Mowbray. Charles has about 2 decades of experience. He has been leading the lead optimization group in drug discovery at DNDi since August 2011. With a distinguished tenure at Pfizer Worldwide Research and Development, including roles as research fellow, he has successfully delivered projects across diverse disease areas, with 2 molecules completing phase 2b clinical studies. It's a pleasure to have him. Dr. Charles, please. Are you able to pull up the slides?
Now you can hear me.
We can hear you now.
Yeah. So thank you, Monalisa. Thanks to the organizers for organizing such a really interesting meeting. It's a privilege for me to follow such a distinguished lineup of experts and to hear so much already about AI. I'm not an AI expert. I'm a drug discovery scientist. I work on neglected diseases and I use AI as a tool. So I hope to give you some insights into how we're doing that. If I can take control of the slides, there we go. So I'll really introduce a little bit about DNDi and our approach to this space and the challenges in particular about using artificial intelligence and machine learning on neglected diseases and how we're trying to address some of those and show you some examples. So DNDi was set up at the request of organizations such as Doctors Without Borders, MSF, because many patients suffer from terrible infectious diseases, often called neglected diseases, for which there isn't a commercial incentive for the traditional pharmaceutical industry to develop new medicines. So shockingly, more than 1 billion people in the world face these terrible diseases and are impacted by these diseases. And yet typically less than 1% of the investment in R&D for new medicines is directed at these diseases. So DNDi tries to address this imbalance, and we're also trying to do this in a way where we can bring the same advances and the best science that we've heard talked about for commercial diseases and bring those to bear for neglected diseases to help our patients. So how do we do that? We're a small organization, but we work through partnership. So we engage with patients, healthcare workers, ministries of health, universities, companies around the globe. Around the globe, with a common interest in trying to address these challenging and horrifying infectious diseases. It's through access to new technologies and working with great scientists in these different institutions that we're allowed to advance our projects. So including in India, but also in Brazil, across Africa, Europe, America. We have partners everywhere, and we're always looking to add to our partnerships and bring new science in to help our projects run more quickly. What are the diseases that we work on? Well, you may have heard of some of these, you may not have heard of others. So neglected diseases and diseases that affect patients in a disproportionate way. So some patients are still neglected even where there's a treatment available. That's the focus for our work. These are parasitic diseases caused by microorganisms. So the kinetoplastid diseases here, some diseases caused by filarial worms, fungi, viruses, and other things for us to think about in the future. There's no shortage of challenge here. But there's some common themes. So these are not really commercially interesting areas. And so typically there hasn't been so much research, and there hasn't been so much investment in research and generation of data. So already you can see that one of the challenges for us is, will there be sufficient data sets to apply AI and machine learning data-driven approaches to all of our diseases? And if we focus just on the microorganism, on the pathogen responsible for the disease, the data set might not be sufficient. So this is a challenge for us, and thinking about how to engage and tackle these diseases requires a bit of mental gymnastics. So as an example, and an example which I'll talk about some more in a minute, it's a little bit different perhaps from some of the other neglected diseases because of the number of people affected by this disease. Dengue is a fever caused by a virus that's transmitted by the bite of a mosquito. It's very topical at the moment. People are talking about the spread of this disease, the impact of climate change, the impact of migration of people to population centers where the mosquitoes are multiplying and infecting people. So huge proportion of the world's population at risk. Every year, close to 400 million infections. And of these, many go on to develop serious disease and can overwhelm the local health care system in the event of an outbreak. So there's a crying need for better measures to address dengue. We think that vaccines will help to some extent, vector control will help to some extent, but we're pretty sure that treatments will play an important part in the armory of dealing with dengue. Similar to the experience of working on COVID, trying to tackle SARS-CoV-2, we think an integrated approach with different types of intervention will be important. The good thing about dengue is there is some data, some in the literature, but it's still not nearly as rich as if you were to look at neurodegeneration, cancer, pain, inflammation, allergy and respiratory diseases. So there's a limited data set. And again, if we were to think just about the dengue virus, as a virus, we're still in a better position than we are for some of the parasites that we work on. But we still don't have so much reported experimental data to really help drive developing models. But for dengue, like some other viral infections, it's not just the virus that's the problem, it's the response of the human host. And so actually understanding the interaction of the pathogen, in this case the virus, and the host, opens up a whole lot more human biology that could be relevant to tackling the disease, much as we've heard in some of the previous talks. So this provides the opportunity for us to think about using AI in a different way for dengue. As I mentioned, we're really trying to bring the best science to our neglected patients, and we've been thinking in our strategic plan about different strands that we can use to help us in this. So both antimicrobial drugs, but also host-directed therapies, not just small molecule drugs, but also monoclonal antibodies, oligonucleotides, mRNA. They may be new and apparently expensive, but we believe that with an appropriate choice of technology and focusing on optimizing the right parameters, it's possible to make these available at an affordable and equitable price for all patients that need them. And bringing much of this together, the opportunity of AI and machine learning to really help us accelerate these approaches. So as we've heard above, and I won't go into this in too much detail, there are some challenges. So the availability of data, which I think I've touched on already, it's not just the availability of data, but it's the suitability of data. So if we're talking about neglected populations or particular populations, are they really represented in the data sets that have been using, especially at a clinical level or at a disease level? Or will there be some bias in the model that means the results of the predictions and the drug discovery endeavor perhaps won't be as appropriate for them as they might be for people that the model was built on, whose data the model was built on? So we have to think carefully about data availability and bias. And I think that touches on the ethics as well. So we want to avoid increasing the neglect by just using the data that we can get our hands on, which may not be as appropriate or as robust for our patient populations as we would like it to be. We don't want to work in a black box. We're working in a place where we're spending the philanthropic money and money from public sources to try for the public good to develop new treatments. We want the work that we're doing to be explainable. We don't want a black box effect. And we really think that the interface between these new AI tools and human scientists, if you like, as a new team, is even more important, and especially so in the NTD context. Although AI is booming massively, it doesn't necessarily mean that AI is booming in neglected diseases, and we need to make sure that we can attract the right talented scientists and computer scientists, data managers, modelers, to apply their skills on neglected diseases and help us here as well. So to come to 3 examples for DNDi, and why are we able to apply these on neglected diseases? Well, the first 2, it's because we've generated vast amounts of data. And for the last one, it's because we're using the published biological literature about human disease that allows us to apply the methods. So the first of these, what I've called parasite painting. In Chagas disease, a disease caused by an infectious agent, a trypanosome parasite. The parasite has an intracellular life cycle, and we can screen for compounds that kill the parasite inside a human host cell. When we do this, we may find new molecules that have an activity against the parasite. They may do that selectively against the host cell, even better. But we've screened already a lot of compounds, and we already know about some classes and mechanisms of action. What we desperately want is new classes and new mechanisms of action. And rather than having to go through a long, slow, and laborious process of deconvoluting the mechanism of action of hits from a phenotypic assay like this, we're trying to use an AI approach, what we've called parasite painting. So capturing images of those parasites in the cell as they respond to treatment with drugs, and looking at also in staining of the cell and the parasite with different dyes, capturing a huge amount of data at different different wavelengths on different channels, allows us to build a fingerprint of the interaction of the drug with the parasite and start to cluster using machine learning and suggest a mechanism of action to see whether it's new or novel. Is it the same as something we've seen before or different? So a very exciting step forward. Our antiviral drug discovery consortium is an AI-driven, structure-enabled antiviral platform trying to develop broad-spectrum antiviral drugs that are resistance resilient. It's doing this at an incredible pace. So this project has just been around for 2 years, and yet already is delivering late lead compounds with terrific profiles. It does this by integrating knowledge, bringing teams together, and using tools such as synthetic chemistry prediction, target selection and prioritization, and many other elements to the process to go at lightning speed. And the last one, it's already been touched on by 2 of the speakers before. So we're working with Benevolent AI in the UK. Benevolent demonstrated the power of large language models and knowledge trees to repurpose in a directed way drugs for the treatment of COVID, and successfully proposed baricitinib. We recognize that we don't know so much about our infectious diseases, but in the case of dengue, looking more about the pathology that is kicked off by an infection by dengue virus, we were able to identify some pathways and hence some drugs which interact with those pathways that we thought could be interesting. And we've been testing those. And at least one of those classes looks really exciting in the models that we have. So we'll be excited to publish some more of this data soon. But what we hope is that this work can quickly lead to a new clinical trial with a repurposed drug for dengue. So really exciting acceleration of our work. In summary, I mean, I think I've showed you we're already trying to use AI and machine learning in neglected diseases research. I do think it's bringing improvements in efficiency and accelerating R&D, but I think we need to be pragmatic. So there are hundreds or thousands of companies developing new tools, and until we know they actually help, we have to be a little bit careful and not get caught up in the hype. So do well-designed experiments, run controls, make sure that we're actually doing something useful. I think there are many challenges that remain in this space in AI, but also in neglected diseases, and we really welcome new partners and suggestions on how we can tackle them together. And I'll stop there. Thank you very much.
Monalisa, you're mute. Please unmute.
I had to do this. So thank you so much for bringing the neglected disease perspective to today's conversation. And in fact, there was a question in the chat session, how could AI help in target identification? And by giving the example of parasite painting, that is one exemplar how we could go down to new pathways or even targets for initiating a drug discovery program. So, and also an example of repurposing. So I think that brings us to the close of expert sessions, but we have all the experts here today, and I really have, I think, 3 minutes. So what I had promised is we would like to have a 30-second view where you feel in near term AI would have the maximum impact. We all feel hopeful, and we have learned that there are really a wide variety of areas where it could make a difference. But in your mind, what do you think would be the biggest significant impact, especially when we keep drug discovery as a centerpiece? So maybe we just start with Professor Sastry.
Yeah, actually, drug discovery is in the domain of medicinal chemists and pharmacologists for a long time. And when biologists come into play, it has become much more complicated. Genomics, proteomics, the data is increasing tremendously. And we are having a lot of data, the next generation sequencing and the personalized data and pharmacogenomics. So the humongous amount of data has become very difficult for us to comprehend with the conventional approaches. So the machine learning, data-driven approaches have become indispensable, whether we like it or not, to make some sense out of this huge amount of data. So now this is the necessity. It's not that we can do something great. We have to use something more than the conventional statistical approaches, like large data analytics has become absolutely important to do something in this space. This is what I see.
Thank you so much. Professor Ahmad.
Thank you. I echo the sentiments expressed by Professor Sastry. Of course, AI is going to become kind of a routine matter. I mean, every science will be done through AI, and that is already there. New areas of AI actually are coming up. The AI as machine learning and as algorithm, like graph-based methods and all, they are already there and widely accepted. Large language models and their derivatives, generative AI, and generating molecules from scratch and synthesizing them afterwards. So many of these areas, I think, will be impacted by the arrival of AI. Pretty much every field of drug discovery will be impacted, and AI will be adopted in every area of drug discovery process, and new areas will emerge in not just the areas which are conventionally pursued, but new areas, new questions which have not been thought earlier will come and will be addressed by AI.
Thank you, Dr. Ahuja.
Yeah, I mean, again, I think I agree with both of my former speakers, but I also would like to say, I think the biggest thing, there is no doubt like the AI will be the part of drug discovery, each and every subcomponent, submodules and sub-submodules. But the biggest issue which I see is the lack of sharing of data between big pharma giants That's the first classical issue. The second issue, which I personally see, is the authenticity of the data. Like as I was keep on mentioning, building models is not difficult. Building accurate models is the most daunting task. And I think these are the two key bottlenecks. We have to solve it if in case we need to basically want to really accelerate the discovery. So these are the two major challenges we need to overcome. Yeah, here I stop.
Thank you, Dr. Mowbray.
Oh, thank you. I think I'd agree with things that have been said before, but maybe two things. One is I think the paradigm shift, so moving to teams of human and machine scientists working together and following a more data-driven approach really changes how teams work. I think that will be hugely significant. And I'm excited also to see how AI and machine learning, which if you can connect to the internet, you can contribute from anywhere in the world. So I think it's a democratizing tool, and I'm looking forward to seeing more researchers engaged around the globe in the tackle on neglected diseases.
Fantastic. I'd circle back to Radha.
Thank you. It's been a really interesting set of talks. I think most of the points have already been covered by my by the speakers. I think two or three things from my side. One is access to data and quality of data. This is a major challenge. The second is we really need interdisciplinary teams, people who understand, even those who are trained in AI ML need to learn something about the biology or something about the chemistry. So truly interdisciplinary learning is very, very important for us to harness these tools well. And the third thing is, I think as the field grows, there is this there is a certain lack of focus on how we are going to integrate AI into all these different disciplines. Do institutions now does every institution now invest in bringing in those experts? How do we drive collaborations and really make AI/ML work for problems like the kinds that Charlie talked about, and also, the AMR example that I gave? How can we harness tools for such immediate, urgent, pressing problems which industry will not touch? And yet our institutions don't necessarily have all of the people or the tools. So I think these are the pressing problems that we need to address.
Thank you so much. And as a product developer, I cannot not say I am extremely excited about what modalities of new line of products it'll throw up. What can we leverage as multiple data set, which I don't think manually we could, and the multi-parametric nature of the analysis. Will it work for everything? I don't think so. It never works. Nothing works for everything. But even if it solves a certain percentage and it grows and we learn and we improve, I think we are making a difference to the discovery process. So I think, I hope our audience felt there is a lot of work that has to be done. I take some of the things that have been voiced, a lot of cautious validations need to be done so that we really build medicines that are safe, efficacious, and at the right doses. That's our simple objective as product developers, but it takes us 15 years to reach that. So the only job I have is thank you so much. I would like to thank the organizers at DNDi, Kavita, for giving us this platform to discuss this important topic, and over to you.